The next speaker is coming from the University of Toronto, Dr. Juan Zúñiga-Pflücker, and he is professor and chair of the Department of Immunology and senior scientist at Sunnybrook Research Institute. He's also a distinguished fellow of the American Society of Immunologists and has served as Councillor, Vice President, and President of the Canadian Society of Immunology. His research centers on the study of hematopoiesis, Notch signaling, thymus biology, and T lymphocyte lineage commitment and differentiation, with the focus on developing model systems for the study of human T cell lymphocyte development from stem cells and the generation of T cells for immune generation and immune regulatory therapies. Today, his title is From Stem Cells to T Cells, Applications and Implications. Yeah, get this for you.
It's going to be there.
What's that? Yeah. There we go. Thank you very much.
Well, it works. All right. Thanks, Joe, and thanks, Norb, for the invitation, and look forward to the rest of the day. So what I'll share with you today is our work on developing both mouse and human models of T cell development and obviously how they can be applied for different screens or analysis or immune regeneration. The work is done primarily by Ashton Troutman Grant, a really wonderful former graduate student, and research associates, Mahmoud and Patricia. My disclosures are listed here about Notch Therapeutics. And what I'll begin is to get us all on the same page. We've just been listening to a great talk by Oliver about B cell development. So I'll refocus you on T cell development. And as we all know, it all happens within the thymus. And this slide is really just to make a couple of points. One of them is to illustrate the need to understand the geographical location aspects that are important for T cell development, where you have progenitors entering from the bone marrow in the corticomedullary region, away from the cortex and the medulla, and then they travel throughout these stages as they undergo lineage commitment to the T cell lineage and undergo rearrangement, and then travel from the cortex to the medullary compartment to undergo final maturation and selection against self-activity and final maturation. So that's one point to illustrate the complexity of the developmental process. And in the bottom is to illustrate the number of cell divisions and proliferation and likelihood of things that can go wrong that take place during early T cell development. You have the HSC coming in from the fetal liver in fetal development, or the bone marrow in adults. And these cells need to undergo a step of differentiation prior to entering the thymus that we usually think about cells that are not HSCs, but lymphoid progenitor cells entering the organ. And those cells are far and few in between. They're basically a handful of cells that enter the thymus on a daily basis. So it's a rare event. But what happens after entry, as the cells encounter these stromal antigens, undergo in the first few stages of development as they commit to the T cell lineage, about a 4,000-fold expansion. So this is a huge number of cell divisions. And following TCR beta rearrangement, undergo another 250-fold expansion. So one incoming progenitor cell will undergo about 1 million cell division expansion to give you 1 million cells. So there's a lot of cell division and things that can go wrong, and having compounds and things that may interfere with that developmental process can lead to dramatic alterations in T cell function and development. So what we wanted to do in the lab is understand how these process takes place and sort of model it and to simplify it as much as possible. So what we knew in the field for a while back was that if you want to model lymphocyte development, hematopoietic development, one easy way to do that was to develop cell-based systems. And the one that we chose to focus our attention was the OP9 stromal cells. And this can be done with MS5 cells or S17 cells or a whole bunch of different stromal cells that can support the development of bone marrow progenitors, fetal liver progenitors, or embryonic stem cells, or pluripotent stem cell, KDR, FLIC1, or CD34 positive cells. You can place these in culture with the right cytokines to support the development of B cells and K cells, or innate lymphoid cells, myeloid lineage cells, erythrocytes, and we can get everything you wanted to out of these cultures. But what you couldn't do originally was make T cells. And to do that, and to study T cell development in vitro in a tractable fashion, you would have to take those progenitors and place them onto a fetal thymic organ culture. And those three-dimensional structures would support T cell development. So what we asked a few years ago was, if you knew what that was the key element that made the thymus special, then we could then take that information and modify the stromal cells to make them more like the thymus so they can support T cell development in a less complicated three-dimensional organizational structure. It turned out that that pathway, that magic, I guess, of the thymus is predicated by the Notch signaling pathway, where you have incoming progenitor cells coming into the thymus, encountering thymic epithelial cells expressing key Notch ligands, in this case, delta-like 4. And the way that the pathway is set up to work is that the incoming progenitors, basically all hematopoietic cells, express Notch 1, but the encounter of high levels of delta-like 4 in thymic epithelial cells, these cortical epithelial cells that are encountering the incoming cells, leads to the engagement of the receptor, which then undergoes a pulling force by the delta-like 4 that is under constant endocytosis in the signal sending cell. And this pulling allows this receptor to be changed in structure. It gets unsprung, and that unspringing of the receptor allows for metalloproteases to cleave it, and then enables the receptor to then act as a nuclear transition factor that goes into the nucleus and acts on a DNA binding protein called RBPJ in mice, or CBF1 in humans. And this DNA binding protein on its own doesn't do much, but when Notch is able to be liberated and transit to the nucleus, it will activate transcriptional programs, in this case, the program to turn a progenitor cell into a T cell. And the way we took advantage of this is to take delta-like gene and transfect OP9 cells that normally would support everything else but T cells. And by enabling then this OP9 delta expressing cells to behave more like the stromal cells in the thymus, to allow for progenitor cells to receive those Notch signals, which are key for the production of T cells. And the sole provision of Notch ligands in these culture systems were able to replicate thymic function and yield T cells, both early T cells and mature T cells in vitro. And you can see that here you have a hematopoietic stem cell coming into the cultures, and then you have the stromal cells expressing delta-like 4, and then having that intimate cell-cell contact interaction that leads to the pulling force on those receptors to enable them to be cleaved and going to the nucleus to activate that T cell program. So these cultures are never just static. The cells are moving around and doing stuff. So what we were able to show thereafter was that you can apply this to human umbilical cord blood derived hematopoietic stem cells. You can culture them in vitro and generate both progenitor cells and T cells. And one application that we had early on was to generate progenitor T cells that could be useful to facilitate the reconstitution of the T cell compartment, both in humanized mouse model, and we're setting up to do this now in a trial. And also, if you have these culture conditions, you have basically a window of opportunity to modify the cells for immunotoxicology purposes to affect their development and see how that could affect the progression, differentiation, maturation, function of developing T cells in vitro. What we went on to show was that these cultures can support T cell development from hematopoietic stem cells and from pluripotent stem cells as well, and generate functional T cells. We can make mostly CD8 T cells that are functional. We're still working on how to best make CD4 T cells using this approach. And then we are able to show that early on, as the cells are being instructed to become T cells, that they become very powerful cells that can home to the thymus and facilitate the engraftment of hematopoietic stem cell derived cells into the thymus. And I have one bit of data to illustrate that here, where we are taking hematopoietic stem cells from a cord blood sample, isolating CD34 positive, 38 negative cells, lineage negative cells, growing them on OP9 delta cultures for about 10 to 12 days. And you can isolate cells that are beginning to go into the T cell lineage because the 34 positive cells gain expression of CD7, which is a marker of T cells in humans. And those early T cell developmental cells are then transferred onto immune deficient animals as our hosts. And these are not SCID, common gamma chain deficient animals that are then given these cells. They're not irradiated to ensure that we're allowing the homing to be as rigorously tested as possible. What we ended up needing to do to make these experiments a lot more efficient was to give the animals human IL-7, a complex with an anti-human IL-7 antibody to facilitate the bioavailability of human IL-7 that mouse doesn't cross-react with the human, as these cells are lymphocyte progenitors and are in need of IL-7. But if you do that and you wait only about 3 weeks, you can see that the thymus of these animals becomes very highly engrafted by the human cells. Over 90% of them are coming from the in vitro generated progenitor T cells. Again, these are the cells that are rare in normally going to the thymus, but now we can produce them in large quantities to enable the engraftment of these animals. And then looking at these cells in more details, you can see that they are, in majority of them are CD4, CD8 double positive cells, as typically one sees in the thymus. And if we look at these cells that are CD4 positive, CD8 negative, or CD8 positive, CD4 negatives, these cells are mature T cells as they express high levels of this TCR complex on the surface. And if you take them out, they're functional, they can respond to mitogenic stimulation, and they will proliferate and secrete cytokines, interferon gamma, and the like. So they're able to mature within this mouse environment, where you can then basically model and affect human T cell development in a more tractable way, in a faster kinetics as well. So what we wanted to do is basically get away from trying to make cells on a two-cell system where you have the stromal cells supporting T cell development, and make it even more tractable for screening or for clinical applications, to make it in a way that it was not cell-based. And this work again was done by Ashton and Mahmoud. And here is Ashton getting a lesson early on in the project about what he needs to do to make this work. Basically, what we wanted to do is to model this whole pathway again. The delta and the notch are critical for making T cells. Without either, T cells are not made, and the thymus cannot support their differentiation. So we know the thymus is great for all of this. We knew that the stromal-based system can allow for development, but of course, most of these stromal cells are mouse-derived, so they're not xenogenic free, and they're hard to expand in large quantities because they're adherent cells and they're difficult to work with. Plate-bound systems can work, but they're not as effective as the stromal-based systems in turning on the pathway. Soluble ligands are actually inhibitory because they coat the receptors and don't engage on that mechanical pulling that is required for the activation of the pathway. So we thought of creating particles that would be able to present the delta-4 molecules in a way that can be inducing enough force on the receptor to allow it to act on the pathway. And the way we went about doing that was to take delta-4 Fc fusions and coated different particles, beads, and different sizes to see whether they would be able to turn on Notch activation. And the way we tested Notch activation was by using a luciferase reporter system that was testing for the Notch activity in these cells. And you have the reporter cells in vitro plated with a control antibody or nothing at all, beads that are unconjugated, or plate-bound delta like 4, which can give you a minimal signal as the cells move over the ligands, they get pulled on and the receptors activated. So we knew that we can detect signal on this approach, and we wanted to know if we take beads of different sizes and we coat them, would this work? And we focused our attention on nanoparticles for many years and end up not having any luck. So all these yellow bars here are nanoparticle size beads, and none of them would activate the pathway. Beads that were very big wouldn't work. Beads that were about 1 micron in size wouldn't work, but about 10 to 20 microns in diameter was the right size that in conjugation with the delta would enable enough force to send a signal to these cells and get activated. So now we thought we had a way to be more simple about how we deliver these Notch ligands to the, in this case, hematopoietic cells, by then complexing the system with a more directed linkage between delta-like 4 with the Fc fusion with the biotinylation region, so we can link it directly into the beads and have it in a way that it was a directional engagement on the hematopoietic cells. And these beads then were tested both with, first with mouse hematopoietic stem cells to show can it work and how many beads to cells do we need. And these are the different ratios set up here with day 7 analysis using hematopoietic stem cells from the mouse marrow. And then you can see that in the unconjugated beads, looking for the presence of early T cell markers in mouse being CD90 or Thy1 and CD25 as an early double negative 2-3 stages, that in unconjugated beads, you don't see any of these cells. You see some NK cells growing, but no T cell development. And when you start putting beads, you start seeing a large number of early T cells. And what you also see in these cultures is the absence of other lineages that are typically able to grow when you don't have Notch signals, like myeloid lineage cells are gone, and B cells, CD19 positive cells, also are no longer supported when you have the Notch pathway turned on. And the later cultures, you can then see the appearance of both CD4 and CD8 expressing cells that have T cell receptors on the surface. So they're becoming T cells solely by the bead-based Notch ligand interactions. Then we ask, can we do this with human stem cells? Again, using cord blood-derived 34 positive, 38 negative cells, cultured with supporting cytokines and comparing them to unconjugated beads over time, starting with the 34 positive cell. You can see that it doesn't take long for the cells to respond to the Notch ligands, and within a couple of days, you begin to see the appearance of CD7 positive cells, which then expand over the time period and begin to give rise to 34 negative CD7 positive cells, which gain the expression of CD5, another marker of T cell development that appears later in development, and later on, even the appearance of CD1A. We compare these bead-based, more 3D dimensional cultures where the cells are engaging those particles with simple plate-bound delta-like 4, which can support some T cell development, but doesn't seem to be as robust and as effective as the more three-dimensional or bead-based system. And then we ask, well, can these cells do what we want them to do? Are they going to generate functional progenitor cells that can engraft and home to the thymus? Can they make T cells? And here we're testing and observing their expansion over the first 2 weeks in culture. So they are exponentially growing once they become T cells. And then we would isolate the 34 positive, CD7 positive cells from these cultures and place them into mice and see whether they can engraft like the ones that we could make from the OP9 delta cultures that are akin to the ones that are the earliest thymus progenitor cells in the human thymus. And much like the cells from the cell culture system, the bead-based progenitor T cells, once given to animals, will show their presence after 4 weeks. And a lot of these cells are human-derived hematopoietic cells in the thymus. Again, majority of them being double positive cells with CD4 and CD8 single positive cells present in these thymi, and not the other lineages like myeloid or B cells in the thymus of these animals. And after a few more weeks, if you look in the periphery of these animals given T cells, or progenitor T cells, you'll see human T cells as CD4 positive, CD8 positive cells, that express the alpha beta CD3 complex on the surface. And if activated through T receptor cross-linking, you would see that they would express TNF alpha or interferon gamma, showing that these, again, like I pointed out earlier on, these cells are mature T cells that can be activated and functionally tested that way. So then we ask, well, if we wait longer in these cultures, can we begin to develop T cells? And you can see that by about a month in vitro, you see the presence of CD1A positive cells, which are later developmental stages, which include the presence of CD4 immature T cells, as well as double positive cells. And later on, you begin to see the presence of some TCR-bearing cells within the CD8 positive stages. So it's not the most efficient way to making a T cell, but at least we know that we can get all the way to a mature T cell. And when you isolate the CD8 single positive TCR-bearing cells, they are, again, responsive to activation. And then we wanted to know, well, that's using hematopoietic stem cells from cords or bone marrow, peripheral blood-derived cells as well. Can we do this with pluripotent stem cells? And to do this, we worked with Gordon Keller to adapt the protocol that they had developed to taking pluripotent stem cells, generating embryoid bodies with the addition of all these different factors to generate CD34 positive cells in these day 8 cultures from these embryo bodies, and then taking the 34 positive cells that are negative for CXCR4 or CD73, and then placing them in vitro and showing that those cells that are immunogenic endothelial in nature will respond to the bead-based delta signals to give rise to T cells over time. So now you see the presence from 34 positive cells gaining CD7 in 7 days and 14 days from the PSC-derived CD34 positive cells. And if you wait longer in these cultures, they begin to acquire the expression of CD5, CD7 positive cells, and go, as humans do, first express CD4 as they become double positive cells, and then mature further to generate TCR-bearing cells in these cultures. So it takes about a little longer because they're PSC-derived, not hematopoietic stem cells, but within a month and a half, you can generate mature T cells in these cultures. You can accelerate the process if you basically, instead of beginning with a pluripotent stem cell that used to be a fibroblast, you can begin with a T cell that is reprogrammed to become an iPS. So it has already a TCR that is previously rearranged, and take those cells and re-differentiate them back into T cells in vitro. And these are the T cells that are iPS as opposed to regular fibroblast cells. Although early on they look very similar, you can see that the former T cells express the T cell receptor much earlier in development than the ones that are undergoing rearrangement for the first time, as opposed to having a pre-rearranged TCR, and show faster kinetics of differentiation and TCR expression. But again, this could give you a way to have a known reactivity as part of the cells that you're differentiating in vitro in these cultures. So with this approach, I think we have a way to make T cells as I wrote here, a remarkably easy way to make T cells if you just have a bead-based system and your stem cells. And all you need to have is a correct bead size with delta like 4 and plus stem cells, you make T cells. And the one bit of advice that Ashton was getting was to use plastics or polystyrene beads to make these delta 4 constructs. And I think you can take advantage of this approach, making T cells from stem cells, modifying the stem cells. You can obviously give them T cell receptors, or as I showed you, or chimeric antigen receptors to target any specificity you want to make T cells for. And of course, you can also take the bead-based approach and generate different additional constructs added to those beads to make it a more complex environment for those cells to develop in. And we've already done that to increase the adhesion and interaction and other factors that you can give those developing T cells in this very controllable bead-based approach. And you can also make progenitor cells, not just T cells, that can be used for eventual immune-regeneration approaches from these cultures. And of course, given the simplicity of the non-cell-based supporting aspects of this approach, that you can adapt it for gene or compound screens as well. And what I'll share with you is a very short vignette about using these approaches to sort of try to understand human T cell development in a little more details that sometimes eludes us when we think about mouse T cell development as a way to illustrate what happens in humans. And one way to illustrate this point, is to look at the early events of T cell development, both in mice and humans. And as I pointed out, it goes from a double negative cell that expresses CD4, CD8, and then becomes double positive, rearranges, and selects. In the mouse, we use these markers, as I used in the talk, CD44, 25. In humans, we're using 34 and 38 on 1A and CD7, so they're a bit different. And one thing that we knew in the field was, when do you need a TCR receptor checkpoints to take place? And one easy way to do this was to look at mice that had deficiencies in their ability to make TCRs. For instance, RAG1 or RAG2 deficient animals have a very well-known block in development. It's a DN3, and they don't make it any further. That's a clear dogma in the field. The question was, what about humans? When is RAG-derived TCR products important for the developmental progression? And it was hard to know, basically because the approach that we were taking was using RAG1 or RAG2 individuals that had mutations on those genes. So these were severe combined immunodeficient patients that had inborn errors in the RAG1 or RAG2 genes, and we were working with those to see what we can learn about the role of RAG or TCRs in T cell development. And the question was, when do the RAG-derived TCRs are important for human, as opposed to mouse T cell development? We worked this with a great colleague, Gigi Notrangelo, formerly from Children's in Boston and now at the NIH, where he was taking care of patients that have severe combined immune deficiencies. And then we were generating from their fibroblasts iPS cells, which we then were differentiated, as I pointed out, through embryo bodies onto cultures to make T cells and ask, where is the defect in their development? A lot of these patients were either SCIDs or Omen syndrome patients that had known mutations in RAG1, and there are multiple mutations. And what we did was to culture these cells from the PSCs in vitro, and then over time, in comparison to the control cells that come from a healthy individual, where you see that the appearance of CD4 and CD8 double positive cells over time, plus the presence of a TCR on the surface, the confusion was that when we were looking at these SCID or Omen syndrome patient cells, some of them would show early CD4s and some double positive cells early on, and then maybe go away or remain in these cultures. And we weren't certain what was the main mechanism for these, because the RAGs were themselves hypomorphs or severe hypomorphs, and we couldn't see the TCR on the surface very well. But if you used high throughput DNA sequencing for the TCR beta gene or TCR alpha, and then look for the rearrangements between J and DJ segments to V segments, you can see that the control cells had lots of rearrangements, and the kids-derived cells that were SCIDs or Omen had a paucity of rearrangements, but they were still there. So TCRs were still being generated, and that could account for some of these developmental defects we were seeing in these cultures. So we weren't certain from this initial work that we published about when exactly was RAG1 and the RAG1-derived TCRs important for the developmental block in humans, because it was hard to just have a full null. So we thought, well, let's make our own. So we took pluripotent stem cells, and we went ahead and decided to ensure a full loss of RAG activity, not rely on compound heterozygotes to look at RAG dysfunction, and generate cells that basically had a targeted mutation of the RAG2 gene locus. It's a very easy gene. It's a 1 exon gene. It's 2 exons, but 1 encoding exon. And then just get rid of RAG2, had different cell lines that we grew as clones, and they had big deletions in RAG2, so the gene was completely gone. And as you all know, RAG1, RAG2 formed this heterodimer complex to cleave DNA to enable VDG recombination. And then we grew the cells and asked, well, now that we know for a fact the RAG is completely gone, there's no RAG activity and no dysfunctional RAG activity, where is the block in development? And early on, by the first few weeks, the cells begin to differentiate much like the wild type cells. They undergo CD7 and CD5 expression as expected, and they begin to show some CD4 development, and also had very similar proliferation and survival differentiation as the wild type cells. So having no TCRs was fine for their early development. But then when we looked a bit later and contrasted them to the wild type cells that undergo CD4 and CD8 double positive differentiation with surface expression of a TCR and intracellular expression of CD3 and TCR beta, the surprise was that the knockout cells looked the same. They had CD4, immature CD4 cells, and double positive cells, just like the wild type cells. So again, one would think back of what I told you about what we know about the mouse studies. This shouldn't happen. The mouse tells us that they shouldn't become double positive cells at all. And yet human cells don't always read the textbooks. And then you have these cells perfectly present there without any TCR on the surface or intracellularly. But yet they're T cells because they have CD3 inside of them. So they are developing as expected. They have no rearrangements. And later on, you can still accumulate as well as the wild type cells. So this notion that differentiation is part of what the TCR beta chain does during mouse T cell development doesn't seem to apply to human T cell differentiation. But there are other aspects of beta selection, survival, proliferation, and opening a whole gene programming at the double positive stage in the mouse context. Is that true in humans as well or not? And so we asked that in more detail, and one easy way is to see what happens later. You reach the double positive stage in the absence of any TCRs, what happens thereafter? And what happens thereafter, the cells are beginning to undergo cell death and they stop surviving, and you can see a big cellularity drop after day 35 in these cultures, in that they reach that point in the absence of TCR beta signaling. One would imagine they're no longer as well as the ones that are in the wild type scenario. So there was one clear difference in the absence of TCR that was much like the mouse. So there was one thing that is affected, it's survival proliferation, but not differentiation. So we asked, well, can we ask the same kind of questions we asked in the mouse models? So what if we give those cells back a TCR, beta chain, or an alpha chain, or together? Does that rescue their survival, proliferation, differentiation, not differentiation, survival and proliferation and programming abilities? So we took cells from PSCs, we made 34 positive cells, gave them a beta chain or an alpha chain, sorted the cells that were transduced, and then put them back in culture. And the cells that were receiving only a control construct that expresses CD tomato don't show any expansion, much like the TCR alpha only. It's not called TCR alpha selection because for a reason, it's not what it does the job. But if we give a beta chain pre-rearrange, then we saw this rescue in their survival and proliferation in different clones of knockout cells. So at least the capacity of the beta chain to induce proliferation, proliferation and survival was the same in humans, but not differentiation. What about the TCR dependent programming that the beta chain is supposed to do in beta selection? Here we did this by RNA-seq analysis, and we're comparing control cells with knockout RAG2 cells, different clones, or the ones that are receiving the beta chain. And the main difference that one sees across these RNA-seq, all these red genes here, these are all TCR genes. that are obviously happening in the control cells because they can rearrange and express them. And it's not happening in the knockouts. So these are all, most of them, 90% of these things are TCR genes. The TCR gene that we gave the cells, the beta gene, is shown here. And these are TCR alpha germline genes that are induced, much like in the mouse, post-beta selection, only when we give the cells a beta chain gene. One of the more important analysis that we noticed, and there were about 30 key genes that were differentially expressed in the presence or absence of a TCR beta chain. It's again, very few genes that are, other than the TCRs, that are affected by having a beta chain in human T cell development. And much like the mouse, one of the key genes was our gamma T, which allows for the survival of the differentiating double positive cells. And then we saw also loss of cell cycle inhibition genes and increased cell cycle progression genes, again, showing the dependency on cell cycle differentiation and cell cycle inhibition changes that are dependent on a beta selection event in human context, but now happening at the double positive stage as opposed to the double negative 3 stage that happens in the mouse. So very different in when it happens, but similarly in how it's happening. So that's an important sort of concept to keep in mind. So with that in mind, I think I just wanted to illustrate that little vignette, that how we can use these culture systems to now interrogate human developmental outcomes in a way that we can begin to understand in more detail how that takes place and the consequences thereof. So now we have, I guess, a better view that the mouse checkpoint happens here fully. The DN3 stage is where beta selection is important to enable the expansion and proliferation. Yet in humans, it happens much later, where that checkpoint is taking place to enable that final set of alpha rearrangements to go with beta to then select for and against self-reactivity. So with that, I'll finish, and again, acknowledge the wonderful work from folks in my lab, great colleagues and our funding sources, and all of you for your attention. Thanks. It's time for coffee.
I've got one question while we're waiting to see what comes online. So first of all, I really appreciate the fact that you're pointing out these species differences, because many of us who work with mice for decades realizing now that the human system is actually quite different.
No, I was shocked that they were so different. I didn't expect that at all.
So the one curiosity, as I was listening to your talk, was the importance of the bead size. Can you
right, yeah.
Light on that? That in itself is very fascinating.
Yeah, the bead size is its own quandary and question. We know that when they were too small, they were likely not enabling the correct engagement. And basically, the easiest answer and the most common sense answer that we came up with was going back to our high school physics and thinking about the importance of force that is required. We know that there are about 7 piconewtons that is required to turn the receptor to be unsprung, to be pulled on. So force equals mass times acceleration. So we remember that formula. So if you need that many piconewtons, and you know that acceleration is going to be basically the little beads bouncing around in media, it's going to be the mass of that bead that is going to be key to enable the force that you need to act on it. So we did some back of the envelope calculations, and you know, it turned out to be about the right size. So so if you get too small, you're not going to enable enough force, and basically the cells are going to just walk away with the beads as opposed to the beads enabling that interaction. Could be all wrong, but it made sense, and the formula works.
I'll read my bios on these cells. Have you done anything with that cell line with these cells? developmental?
Yeah, we did a lot of the early work that we did in the mouse actually was using embryonic stem cells on OP9 cells to grow B cells because we couldn't make T cells early on. and those, for the most part, are both types of B cells show up, fetal and adult types. I think depends how you grow your human genetic endothelial cells and what type of cells you're differentiating thereafter.
But your red knockout cell line.
Oh, the rag knockout cells now. Oh, you mean that one?
Is that reproducing mouse or is helping like a pro-B?
Yeah. We only look for absence of BCR. We didn't actually phenotype the full set of markers that you would do. Yeah, good question. It was just to see whether they had any rearrangement capacity whatsoever, and the answer was no. Yeah, we should go back and contrast the well-known staging in mouse B cells, whether that's also seen in the absence of pre-BCR signaling.
Okay, we do have one question here that came in online. I had a question about the timing of the TCR beta selection in the human in vitro system. Is that representative in timing temporally to what would happen in vivo in humans?
Yeah, it's a really good question. And the answer is yes. If we take away the first 8 days as you're making them become hematopoietic, the timing is similar to what you would see in the way we've modeled human T cell development by looking both of in a humanized mouse model at the same timing and based on all the new work that have been shown in single cell RNA analysis from fetal human development and the timing that they can extrapolate from that, it looks the same. Yeah.
So I have one question. In the model where you were using your microbeads to push T cell differentiation, and out roughly 5 weeks, you get mature CD4s and CD8s, albeit inefficient.
CD8s we get really well.
Yeah,
CD4s
yeah.
not so much.
Yeah. So they expand in response to mitogenic stimulus, but I was wondering if they also have normal cytokine production compared to normal CD8s.
They're looking like a TC0. They're like naive CD8. They don't have any bias in terms of which cytokines you're making. So the idea is that you would have to activate them under different cytokine conditions to make them become different types of CTLs. Yeah, they're naive cells.